We now move to the first session speaker, who is actually my co-host, Dr. Vachel. She is a professor and UP scientist of the Animal Biology Division, Institute of Biological Sciences, University of the Philippines, Los Baños. Her research interest is parasitology, focusing on the role of parasites in various ecosystems and their impact on public health. She has authored several scientific articles, book chapters, and laboratory manuals in parasitology. Dr. Parlier is also an officer of the Philippine Society of Parasitology Incorporated, and she obtained her doctoral degree from the Graduate School of Health Sciences in Kobe University, Japan. Dr. Parlier?
Thank you very much, Dr. Cadiz, for your kind introduction. Let me just share. Okay, so screen share. So thank you for this opportunity to share our research results regarding the role of agriculture intensification on the emergence of zoonotic diseases. Okay. So we all know that access to safe food is in itself an element of food security. Okay. So the World Health Assembly Resolution on Food Safety stated that everyone should have the right to an adequate supply of safe and nutritious food, and that there is a need to implement a national and regional or local mechanisms for food-borne disease surveillance. And also, each government should take the necessary measures to ensure the availability of safe food for all in order to sustain the health and economic development of their people. Food-borne diseases are those that are the results of pathogenic microorganisms such as bacteria, virus, parasite, and parasites. No? So these may also be due to chemical substances such as pesticide residues, veterinary drugs, food additives, and many others, which tend to have acute effects on human health. So the problem usually arises due to contaminated food. So the WHO estimates that 1 in every 3 people worldwide suffer from FBD every year, and that 2 million people die every year from severe food and waterborne diarrhea-related illnesses. Most of these illnesses are due to microorganisms and chemical contaminants. In developing countries, in the Philippines particularly, some cases of FBD are not reported, and therefore the true dimension of the problem is unknown. Okay. The Food and Agriculture Organization and the World Health Organization in 2018 released a list of food linked to outbreak-associated diseases. So we can see here in the pie chart that in recent years, outbreaks of foodborne illness linked to fruits and vegetables are on the rise. The fruits and vegetables, we know they are healthy for our well-being, providing essential vitamins and minerals and fibers. These fruits and vegetables once were thought to be relatively free of disease-producing pathogens. But outbreaks have been linked to pathogens such as Escherichia coli 015787, which could be fatal, particularly to immunocompromised individuals. Salmonella were reported from apples, lettuce, cantaloupe, and sprouts. Listeria monocytogenes contaminating cabbage and cantaloupe. Shigella in parsley and lettuce. Cyclospora in berries. Toxoplasma in fruits and vegetables, and soil-transmitted helminths like ascaris, whipworms, hookworms, contaminating also vegetables. Okay. So changes in the dynamics of these pathogens have undoubtedly contributed to the increase of pathogens, no, in the farm. So including the impacts of climate change and other anthropogenic activities, as well as changes in growing, harvesting, distribution, processing, and consumption practices. Contamination may occur at several points along the food chain, in the farm or in the field, during processing or post-harvest, at the market or at the point of sale, or at household level. So the farm is a perfect ecosystem where environment, animal, and human interact. So we call this as the environment, animal, human interface. Farms are potential ground for contamination. Farm produce are vehicle of transmission for microbial and parasitic infection. Some vegetables are eaten raw, and parasites are able to live and infect potential hosts. Despite reports of several outbreaks worldwide, not much has been studied regarding parasite contamination at the farm level. Unfortunately, there is no such thing as parasiticide, which we can apply in the farm. We have insecticide, fungicide, molluscicide, but there's no such thing as parasiticide. Moreover, expansion of agriculture promotes encroachment into wildlife habitat, such as agroforestry, leading to ecosystem changes and bringing human and livestock into close proximity to wildlife and potential vectors. There are several examples by which agricultural expansion and environmental changes were associated with an increased risk of zoonotic diseases emergence. And these are driven by the impact of expanding human population and changing human behavior on the environment. In fact, some studies mention that the rate of future zoonotic disease emergence or re-emergence will be closely linked to the evolution of the agriculture and environment nexus. We have done several research works which are proof of the role of agricultural practices linked to zoonotic transmission of parasites. Now, this is a recent paper we published and available as open access, so you may read them for your information. We also have investigated the extent of parasite contamination in environmental samples like the soil in the farms, in selected organic and conventional farms in the Philippines, as well as examining the freshly harvested vegetables from the farms. We also tested composted manure used as organic fertilizers, and we found that they are also contaminated with parasites. So just to give you a picture, a summary of the results that we have done, of the research that we have done, the prevalence of parasite contamination of freshly harvested vegetables range from 15 to 35%. Higher prevalence was reported during dry season, and higher contamination of vegetables, surprisingly, from organic than in conventional farms. And later on, I will explain why there was higher contamination in organic than in conventional farms. Also, remarkably, leafy vegetables found to be higher incidence of parasite contamination than fruit vegetables like tomatoes or cucumber. So these are pie charts showing the proportion of parasites that we have harvested. We have ascaris. You are familiar with ascaris, which show the higher prevalence. Trichuris, 26%. Hookworms. These are common parasites of animals and humans. Toxocara is a parasite of dog. Tinea and other parasites can be also found in animals and humans. Surprisingly, these parasites that we have harvested or recovered from the farms are also common among our children. In fact, more than 50% of our children in the Philippines are infected with ascaris, trichuris, and hookworms. Now, these are just photographs of the parasites that we have recovered from vegetables, from soil, and other environmental samples in the farm. And just to show you that we have harvested more parasites from organic farms than in conventional farms. Among the vegetables, so we classified them into leafy vegetables and fruit vegetables. Leafy vegetables were found to be more contaminated, with green eyes showing higher incidence of parasite contamination. So this is an example or a photograph of green eyes. So we hypothesize that because of the crevices, then the parasite eggs or larvae may be they may be attached on those crevices, and therefore, if not properly washed, may be source of contamination to consumers. The presence of parasites on fresh produce may be attributed to what primarily come in contact with them. So we tested the wash water used in the farms, the soil, of course, where the crops are planted, the animal feces were also assessed, and we also collected fecal samples from the farmers. And surprisingly, all were found to be contaminated, and all animals were also found positive. All farm animals, even domestic animals like dogs and pets, were found to be contaminated or infected with parasites. So to summarize, the parasites in human fecal samples are from farmers. The animal fecal samples, soil, and wash water use in the farms showed positive association with the incidence of parasite contamination in farm produce. We also recorded some farming practices, and results revealed that open water sources, the use of manure, presence of animals, some sanitation and hygiene practices of the farmers contribute to parasite contamination. So we also did molecular analysis. This is very important to identify the specific species of the parasite and therefore know the exact source of these parasites. So the ascaris was found to be Ascaris suum. Suum is specific only for pigs. Trichuris, we found Trichuris trichura and suis from humans and pigs respectively. Toxocara is Canis species from dogs. Hymenelepis were diminuta and nana from rats. Hookworms were found to be Ancylostoma ceylanicum, which is a hookworm of dogs. Balantidium coli species from pigs. Angiostrongylus cantonensis from rats. Then Echinastoma malayanum from rats, and Giardia intestinalis of subtypes A and B from humans. So we see here that the sources of parasite contamination were from farm animals and domestic animals and also humans in the farms. So the possible contamination sources, therefore, are raw or improperly composted manure, contaminated wash water and irrigation, presence of animals, including pets, rodents, and wild animals in the farms that may encroach in the agriculture setting, lack of field sanitation, and cross-contamination. So we summarize here the parasite transmission dynamics in the food supply chain, starting from infected animals in the farms that may contaminate crops and other animals that subsequently have impact on the production process and the next level of the food supply chain. But human factors such as education, awareness, socioeconomic status, health and well-being, and cultural practices also have impact on this transmission dynamics. So we have to control this at various levels through good farming practices and also therapeutic and preventive treatment like deworming of our animals in the farms, including humans, and then for control of parasites, there should also be a mechanism for surveillance at the regional level or local level, detection and removal of parasites in the farms, and removal of the contaminated food from the food supply chain. So there are several challenges and future directions for elucidating parasite contamination at the different levels of the food supply chain. One is elucidating the role of culture, socioeconomic, and governance, the impact of climate change. We can also do modeling for predictive studies, improve diagnosis using molecular or immunodiagnosis, or even omics studies, and interdisciplinary and interagency approaches are also very important in addressing our questions regarding zoonotic transmission. So there should be a coordinated effort to bring the resources of modern science to bear on the control of food-borne illnesses. And the approach is One Health. If you want to have to address human health, we need to address animal health as well and the environment health. So I'd like to also recognize the efforts of my students and research assistants and funding agencies of our researches. Thank you very much for your time.
Thank you very much, Dr. Vachel Palier. So if you have questions, just type your questions there and will be addressed after the session speakers.